Good morning and welcome, everyone. Please join me in bringing to the podium Chair of the International Board of Directors, Grant Beard.
A little Jimmy Buffett in the morning. Thank you, Elizabeth. Maybe we could have cheeseburgers in paradise later this afternoon. Good morning, everybody. And yes, I am Grant. As chairman of JDRF, along with my amazing partner and vice chair, Michelle Griffin. Where are you, Michelle? We would like to formally welcome you all to this year's Global Mission Summit. We know that many of you have traveled great distances and really made a big effort to join our gathering, so we are so appreciative of the effort made. And hopefully everybody had a good time last night. You know, when I look out onto this group, this gathering, I'm amazed. You know, you all might not think about it this way, but we are the influencers to an affliction. That is quite a responsibility indeed. You know, whether you are a generous donor, an incredible scientist, or work in one of the many organizations that support T1D, we all want the same thing, right? We want to modify, we want to prevent, and ultimately, we want to cure this affliction we call T1D. And if you'll allow me for a moment to sort of switch my hat, I'd like to say thank you to all of you as a father, because I'm really here as Emily's dad. And because of you, Emily now lives in the best control she's ever had because of these amazing devices that adorn her body. But much, much more than that because of all of you. Her tomorrow is so much brighter than her yesterday. So I thank you. This walk we've been on to find a cure has been a long one. And I think Doug would agree with that, and you're going to hear from Dr. Melton shortly. But you know, sometimes on a long walk, you have to stop and turn around to appreciate the progress that we've made. And the progress that we've made has been incredible. And I think that you will hear about that over the next couple of days. The good news, maybe it's great news, that we have really good science. Right? Much of it's sitting in this room. But good science without commercial availability to my Emily doesn't really mean much. So I'll pledge to you, as JDRF, we will continue to work globally. We will continue to work collaboratively with our partners, many of you, again, are here. And we will work tirelessly to do everything we can to impact all Emilys as soon and as at the highest level we can, no matter where they live, in what zip code or what continent. That is my pledge. You know, these events just don't happen. They take a lot of work. And Aaron said this last night, but I just like to reinforce and say thank you to the incredible staff in the United States, and a special thank you to the staff here at JDRF UK. I mean, this is just an amazing program. And I don't know if Alex is here. I had the pleasure of eating last night with Alex Getty, and thank you for allowing us to come on to the grounds of your amazing home. What a phenomenal place to be able to share with old friends and be able to make some new friends. And now I have a favor to ask, but it's not that kind of favor. We'll do that at a different venue. This is your summit. It's not Aaron's or mine, it's yours. So take advantage of it. Get to know the participants, ask a million questions, and have a little fun. And I'm pretty sure when you leave tomorrow afternoon, you're gonna have two things. You're gonna be so inspired by the advances that we've made, and you're gonna say to yourself, man, do we have good people in the T1D corner? So please enjoy your summit. And so with that, I'm going to turn the podium over to our fearless leader, a great colleague, and a dear friend, Dr. K.
Nice. Oh, a little nervous. Alexander and the Getty family, thank you again. You know, this is, we can say it over and over. Some of you know I'm a bird watcher, and seeing the kites fly over the fields yesterday, this is a special place. So thank you so much for hosting this incredible group of T1D champions who are in the room. Thank you, Grant. You are a tremendous friend, a tremendous champion. Chairman of the IBOD, Michelle Griffin, thank you for all you do, our vice chair. I want to thank Jennifer Bennett. Jennifer is our Global Mission Board chair, which is an incredible vehicle that drives our volunteerism around the world. We have an amazing room here, but I do want to say special thanks to David Panzier. I mentioned David as our incredible partner, trustee at the Helmsley Charitable Trust. I can't say it enough about the critical role that the Helmsley Trust plays, and David, on behalf of his daughters, does as a T1D champion and friend. So David, thank you. And again, I said last night, this takes a village. We recognize our One Society members, researchers, so many incredible partners here, coming together, working from different angles, and trying to drive our mission forward. As Grant said, and I said last night, I want to really thank our international affiliates and Karen Addington here in the UK, and of course, with heavy hearts after losing the Queen yesterday. We are so appreciative of all the hard work. And all of you for coming together at the Mission Summit outside of the US. It's really nice to be back traveling. And I'm so excited about today and tomorrow and talking about progress, as Grant pointed out. All you've done, seeing the progress we're making, and hearing about where we're going. So this venue is a testament to our mission, accelerating life-changing breakthroughs and driving to a world without type 1 diabetes. Global organization, uniting people with type 1 across the world to drive that mission forward. Life-changing breakthroughs. And it's because of your leadership, partnership, generosity that JDRF is the leading volunteer-powered T1D global organization. Working across the pipeline, you hear me say that all the time, starting at discovery research, but making sure that research gets into the hands of people, that it's accessible. That over the 50 years of history of JDRF, that we've funded research in over 50 countries now. So where are we now? This is an amazing time. You're going to hear from Dr. Melton, and in our sessions throughout, hear about progress and advancement. And as Grant said, and as I will continue to reiterate, people are doing better because of research. People with type 1 are living healthier lives because of research and because of advocacy. And we can give amazing examples that I'm really proud of. Working with Cynthia Rice and the team in the US, fighting for coverage for CGMs and closed-loop systems and insulin access. I have to give Mike Wilson and the team, Mike Wilson, that's an inside joke, I said Dave last night, despite 17 years of working with Mike. The team in Australia for championing better access to systems in Australia, closed-loop system coverage all the way down to the age of 2 little babies being able to get control of their diabetes. I'm incredibly proud of the work we've done in changing pregnancy outcomes. Right here in England and in partnership with our team in Canada, we funded the CONCEPT trial, which changed the standard of care for pregnant type 1 women across the world. Helen Murphy at Cambridge, amazing T1D champion. The work we did on Medicare coverage. You know, people always say, what does access mean? The pipeline, what is the pipeline about? If you can't afford one of these breakthroughs, your life isn't changed for the better. And gosh, was that frustrating when Medicare type 1 people could not access CGM, the most vulnerable people to severe hypoglycemia. And that fight is was one of the hardest, but gosh, the outcome in getting coverage is one of my proud moments for us and the team at JDRF. So again, I mentioned my brother Steve and me. Some of you know Steve, who's a very successful, super smart guy, but has hypoglycemia unawareness. And I've been in the back of an ambulance with him where I thought he was going to die, and have given him multiple glucagon shots. And now I see him, lives right around the corner as a dad of 2 kids, running his own business, and using a closed loop system and getting low very, very rarely. And that's life-changing. It's life-changing to Steve, to his wife, to my parents, to me, to his kids, and that's all because of folks like you. I mean, that's amazing. So these advancements, reducing highs and lows, that's one side of the equation, but it's also the other side of the equation, life. Life is more than diabetes. And taking some of that burden off of our shoulders, we were just having a discussion, the Rotellas and I, outside, about how there's just a lift off of your shoulders. Now, all that said, I'm gonna take my pump out right now and say, my blood sugar's 238 right now. It's not normal. We're still not there. It's still really hard. And why this organization was founded was to cure type 1. Take devices off, and that's with all respect to my friendly colleagues here who are helping us, but that's our goal, is to move on. And right now is an unprecedented time in science. In type 1 science, but in science in general, in immunology, islet biology, and we're seeing incredible advancements. So here in the room are some of the best. You're going to hear from Dr. Melton in a moment, who will talk about the progress that they're catalyzing in T1D. You can think of it, obviously coming out of COVID, as the tools that are driving medical advancements are different than they've ever been. I think the COVID vaccines are an example of that. The scientific community using these tools to move things faster. And Sanjoy, our T1D Fund team, we're all working with our scientific community to take those advancements and move our fields faster towards cures and prevention. And research like this, when I was in the lab, did not happen so fast. We've seen incredible progress. So now, translating that into T1D, you'll hear from some of the best and the best. And what it means practically for this community is now we have dozens and dozens of potential curative and preventative therapies in advanced stages of R&D. It's not just in cell culture, it's not even just in mice, moving all the way into human trials. Dozens and dozens of shots on goal. So these cell therapies, disease-modifying therapies, are going to happen. It's not a matter of if, it's a matter of when. So how can we unite to make that happen faster? That's what this is all about. Stem cell-derived islet replacement therapy. You'll hear again from Dr. Melton. The team at Vertex is here. To me, holds such incredible promise and hope. The results, as you've heard in the news, the preliminary results are amazing. And the push that we're making to drive towards immunosuppression-free cell therapies are also benefiting from the advancements we're seeing in science. You've heard of CRISPR technology. the idea of gene editing. This is what our centers of excellence have been working on to remove the need for immunosuppression in stem cell-derived islet replacement therapies. My family with two T1Ds worries about our children. My parents now have, I think, this is embarrassing, I think it's 13 now, grandchildren, 5 siblings. And prevention is a key priority for us at JDRF and for anybody living with type 1 because we have a genetic predisposition. For the first time ever, we have teplizumab, the first potential disease-modifying therapy that could be approved in the United States this year, in the next couple months, in November, and I'm looking up at Sanjoy, we're hopeful to see that. And this is amazing. So everything we do is aimed at improving lives and then accelerating towards cures for T1D. Working across the pipeline, from basic research all the way to access, multiple approaches, working with researchers across the world, working with our industry partners, leveraging expertise with funders, supporters, advocates, all leaning in. So within the last 6 years, we've catalyzed a number of new vehicles to try to make that even happen faster, and a number of you have been incredible supporters of that. We have our T1D Fund team here. To me, the T1D Fund is one of the most important things we've done, in my opinion, in the last decade at JDRF. Venture philanthropy, catalyzing additional capital, driving it into the space, taking donor investments into the fund and working with commercial entities that are taking our research and trying to bring them to the people. And what it's done is catalyze an investment market by attracting other dollars, huge leverage. So for the many fund donors in the room, as you know, your dollars are being leveraged by other private venture capital companies and vehicles to multiply our investment and our knowledge base to drive those advancements towards the market. 2016, when the fund was created, there were only a handful of companies investing in T1D, particularly on the curative and preventative side. And now we have over $640 million of investment in the space, alongside ours, that's being used to drive our field forward. We also have our centers of excellence, and I'm looking out on John, thank you, and a number of families for your leadership and philanthropy and taking our centers, and Doug was the lead at our New England Center, that's partnering world-class institutions, incredible scientists, and filling some of the most cutting-edge and innovative research gaps and driving them forward. Leveraging new technologies to make stronger islets, looking at mechanisms to cloak cells from the immune system, as I mentioned, using CRISPR, for example, or innovative ways to encapsulate cells, looking at new immunotherapies. Many of you have leaned in to make this happen, and for some of you who are on the front line, I'm looking at Karen, for example, are seeing how these centers are bringing together just the most amazing minds to solve some of these incredible, challenging problems. And because of these centers, we've raised $23 million to support them, incremental additional dollars flowing into T1D research, with a goal of raising $42 million. We have 5 centers, 3 in the US, 1 in Canada, and 1 in Australia now. So this is all about working the pipeline, research and advocacy coming together, improving our lives today, curing type 1 tomorrow, driving across here in the UK. I'm really proud of the work we've done to improve CGM access. In Canada, fighting for access to CGM in pumps. I mentioned Mike, CGM, flash glucose monitoring, access in the adult population, which wasn't accessible. You know, in the US, we've been fighting particularly hard on insulin access. I mean, how is it possible that in the United States, we still have people paying $350 who are choosing that versus a rent payment? Making sure that private insurance is covering CGM, Medicare, Medicaid, and you've seen that we've launched an initiative with Civica to essentially make insulin available at cost no matter what your situation is. $30 a vial, $55 for a box of pens. Worked on the bill that was just passed by the Senate to cap the price of insulin at $35 a month in the Medicare population. And these are real wins to ensure that people have access to the best care. Now, I do want to point out that there are still gaps, and Grant alluded to this, on access around the world. The burden of type 1 is not just in the UK and Canada and our affiliates. It happens in every single country. And it's particularly painful to see in developing countries. There are hundreds of thousands of people with type 1 diabetes who don't have access to even the most simplest forms of care, insulin and test strips. And the key thing, and I think this becomes really important for our community to appreciate, is the faster that we drive access to everybody with type 1, the faster everybody with type 1, including our loved ones, many of us who are on the front line of care, will have access to cures. Because this market model lags for lack of access. There is a market dynamic that needs to be fixed, and that is at the most basic elements of care and even in advanced countries such as here in the UK. We see these impediments as slowing down progress to cure. So you're going to hear a lot about our global index, our global access initiative, and I think some people worry, well, are we losing focus on curing type 1? No. All of this ties together to accelerating cures for type 1 and ensuring more people want to lean in and partner and work with us, including people with diabetes and the industry who develops the therapies that ultimately we benefit from. So ultimately this system, the kind of thinking of it as a global marketplace where T1D therapies are approved faster and accessible faster, I firmly believe will drive to cures faster. So that becomes a exercise in breaking down barriers, using our advocacy and our voice across the globe. So I'll just finish by saying that T1D is a global problem, and I'm proud of the global nature of JDRF. Proving global access is a key priority of ours to create a thriving market, and a thriving market with great access will catalyze better investment in cures. You are our most enthusiastic supporters, this group. I mean, it's just such a humbling experience to be in this room. But this group is on the front line of leading that charge. Again, just to give you a little preview about the T1D index, an incredible credit to Mike Wilson, Sydney Jovic, Tom Robinson. You'll hear for the very first time, and we'll be launching this publicly in a few weeks. It's a model that allows us to look at how many people with type 1 are in country by country and how people are doing. So what you're going to see is how many people are in our affiliate footprint, but importantly, across Europe, across Africa, across South America, in the Middle East. It's going to be published on September 14th in The Lancet. Diabetes and Endocrinology, one of the most prestigious and trusted medical journals. And data from this index provides a platform for us to significantly ramp up our advocacy efforts and drive better access and improve the lives of people around the world. I think this is going to take partnership. Of course, Helmsley has been a leader on this front. We have our friends from the IDF here. It's going to take, again, a village to really lean in and make a huge difference, but we can. So you're going to hear more from Tom Robinson. I can't wait to be with the team as they do that over lunch tomorrow, and I think it's going to be really exciting. So again, we're one world, stronger, working together. In many ways, to me, this is the most exciting time I've ever been at JDRF. My family's been doing diabetes for 40 years. And I truly believe that I will take off my pump because of all of you. My brother will, and your loved ones will. And again, I'll say it's not a matter of if, it's a matter of when. So because of you, we hold the potential to deliver that and realize that for all of our loved ones and people around the world. And what that means is we have to unite our communities around the world. A more united community of T1D people will create a catalytic, more powerful, more unified voice. And that means donors. Many of you are philanthropic donors. It means advocates. It means the research community. It means policymakers, healthcare decision makers, industry. It means more people having their lives changed for the better, more people working to make those next changes happen sooner. This is the time. If not now, when? So we need your support. We need folks to lean in. You did so much, particularly during COVID, and we need to keep pushing. So we need the partners like the Helmsley Trust, the Morgan family, IDF, researchers, Vertex. We can empower and enable progress and fight T1D faster and better than we've ever seen before. So here we are at Mission Summit. Kind of feels like England, a little rain, sunshine. And you'll get to talk, as Grant said, take time, talk to folks. I mean, I am meeting and speaking, and I learn every day. Researchers, physicians, advocates, all of that's gonna add up to improving lives today and tomorrow. So thank you guys so much for making our vision a reality and driving towards our ultimate goal of walking away. So now it's my real pleasure to introduce Dr. Doug Melton, an incredible friend, an incredible T1D champion, world-renowned cell biologist. He's going to tell his story, so I won't. Again, you've seen on the news, read some of the work that he will again talk about, so I'm not going to steal the thunder. Headed up our New England Center of Excellence, and then gave me a call, and he said, I got some news. I'm going to move over to Vertex, which I think is just a real testament to the potential that we see in cell therapy, and Doug will talk about that. New England Center continues to be a powerhouse and will continue to drive towards those advancements to remove the need for immunosuppression. But Doug, you're an amazing visionary, and I can't wait to hear your update on the progress you're seeing. Thank you. And thanks, everybody.
Well, thanks everyone for coming. I always love listening to Aaron because he sees the big picture of things, and I find it quite inspiring to hear him talk about this. In a sense, I'm going to change approaches and talk about something very narrow, which is just trying to cure the disease. So while he's talking about important issues for global type 1, I'm going to kind of bring you back to your high school or college biology and just think about one thing. Now, as Aaron mentioned, maybe I'll put my first slide on here. That's not it. Yes. I worked for several decades at Harvard University, and I was thinking about that as I was about to speak. In fact, I'll give a lecture there on Monday, that my challenge then is to get the students' attention, usually like shopping on eBay or on Facebook. And I noticed none of you are really looking at your phones or your computers, and that's just a delight to me, because usually I have to kind of tell jokes or pause or something. And so it's wonderful to talk to an audience that actually cares about what I'm going to speak about. So thank you again for coming. I'm also accustomed to being interrupted all the time, so I'm going to show you some science slides, and just raise your hand or yell out if it doesn't make any sense. What I thought I'd do today in the next 40 minutes or so, to reserve time at the end for questions and answers, is tell you about my view about what diabetes is and how I think it can be cured. Now, as Aaron rightly said, there's enormous advances in pumps and insulins, and I don't mean in any sense to discount those. Those are important, but that's not what I think about. And just on the personal side, I might mention, I called my wife last night to tell her, what a lovely place this was. And she said my son Sam had been out running and his pump fell off, and now they were in the woods looking for it. And now she says, of course, on Monday I'll have to deal with the insurance company and everything. And so I'm just thinking, like, wouldn't it be nice to give people cells so they make their own insulin, and then there's no more insurance companies, there's no more pumps, there's no more monitors. And that's really what I think about every day, is not better treatments. I don't think about complications, I don't think about prevention. I just think about delivering a functional cure. On the personal side, if you allow, my own son, Sam, got diabetes when he was 6 months old. And for many of you, you can imagine what it's like for my wife to be breastfeeding a child, trying to guess how much carbohydrate and insulin a 6-month-old needs. And some 10 years later, my daughter, Emily, like Grant's daughter is named Emily, got diabetes. So if I seem a little obsessed with it, it's true. I really just focus on trying to do something for them. And so what I'm going to do today is tell you, as I said, how I think about diabetes, how I think it can be treated. I'm going to talk about work I've done with my lab at Harvard over about a 20-year period, and then talk about what I'm trying to do now with colleagues at Vertex, which I think is the next level, and Aaron has pointed to this, dealing with immunosuppression. But again, please raise your hand and ask questions at any point. And I'm going to move over here so you can see my slides. I was excited to hear there were teleprompters because I've only seen those on TV and I was hoping to try one. But in the end, I'm not going to use that. So as I said, I want you to think with me today about cells as living medicines. This is quite different from thinking about injecting insulin or taking a pill. This is where you give the patient their own factory to make insulin for the rest of their life, essentially make them non-diabetic. And as I mentioned, and I'm on leave from Harvard, but I'm now an employee of Vertex, and they tell me that I have to make very clear that I don't speak for Vertex, and I'm not here to try to tell you to buy their stock or anything. I'm just going to tell you about why I moved there and what I'm doing. Okay, this is my view of diabetes. It'll be a simple introduction known to many of you. There's a picture there on the left of your pancreas, which is about the size of a banana. And you probably know that inside the pancreas, kind of like raisins in raisin bread, there are these islets. You've heard much about pancreatic islets. That's a confocal image of a pancreatic islet. If you're not diabetic, you have about 200,000 to 500,000 of those islets inside your pancreas. So like 200,000 raisins. Inside the islet is the insulin-producing beta cell, shown there stained in blue. The green cells make the other hormone you know about, glucagon, the counter-regulatory hormone. Each islet has about 10,000 cells. So if you're not diabetic, you generally would have about 1 billion beta cells. Now, of course, as you know, in diabetes, those cells are dysfunctional. My view of diabetes, both type 1 and type 2, is it is a defect in the insulin-producing beta cells. That's a somewhat controversial view. In Britain, type 2 diabetes is considered beta cell insufficiency. In America, type 2 diabetes is considered a problem with insulin resistance, from which you should conclude we don't really understand it. Type 1, on the other hand, is a consequence of the destruction of the beta cells by autoimmunity. And I know many of you know that. I thought I would just set the stage with, this is how I think about the problem. It's all about the beta cell. So there's a little trick picture, in a way, of what happens in type 1. You see the autoimmune system, for reasons no one understands, decides to kill the beta cells. Sort of an interesting problem, if you like. Why doesn't it kill the alpha cells next to it? We don't have any clue, really, about why that's the case. We do not understand what's special about the beta cell. This is well known to all of you, and this is essentially what I want to forget about and not think about anymore. I show this picture. I had the pleasure last night of sitting next to the former CEO of Becton Dickinson and was teasing him about syringes. I don't ever want to see another syringe again. There have been enormous advances because of the JDRF in insulin pumps and continuous glucose monitors. And in no way do I want to delimit or insult all of the activities of people who do that. That's a really important advance. I just want to get rid of it. I want you to think about what I'm going to talk about like moving from gasoline cars to electric cars. I want to get rid of all of this. I also like to get rid of the insurance companies, which I don't know how to do, but my wife spends an enormous amount of time dealing with the craziness of just trying to get supplies and get reimbursement. And we are a reasonably educated family of quite means. As Aaron said, I can't imagine what it's like for people in other countries. Okay, so this is what I want to get rid of. Is there a better way? I'm going to make the argument that there is a better way, and I'll tell you why. Beta cells read blood glucose every millisecond. That's 1,000 times a second. The best continuous glucose monitors read blood sugars, they claim, every 5 minutes. I actually think it's closer to every 15 minutes. In addition, the beta cell releases not a bolus of insulin, but just the right amount of insulin. It squirts out a tiny amount of insulin to control blood sugar. So you might think of this maybe like, say, the thermostat in your house, where you have a heater and an air conditioner. Your thermostat is reading the temperature, and as you know, the heat or the air conditioning will come on every few minutes. The beta cell is the opposite. Imagine a heater and an air conditioner that was reading things every millisecond and controlled the temperature at 20 degrees centigrade, since we're in England, or say 70 degrees Fahrenheit, without any variation. That's what the beta cell does. So I would suggest that if we can make beta cells, that's nature's solution to the problem. The beta cells are missing, so let's make more. So I think of the challenge for type 1 diabetes in a simple-minded way. There are two issues. There's the loss of beta cells and the presence of the autoimmune attack. Make more beta cells and figure out how to manipulate the immune attack. I've spent more than 20 years on the first thing, which is what I'll tell you about today. And someone last night asked me, a very nice person said she didn't really understand what a scientist does or how a scientist thinks about things. And I was thinking about that last night. It's a hard question to answer. So I thought I would just tell you about how I think about the problem. So we'll go into a little science detail now. And it involves the challenge of discovery, which the JDRF supports so well. Discovery means you don't really know what's going on. You're kind of fumbling around in the dark trying to figure out what's going on. And so I had the pleasure of being educated here in England some years ago at Cambridge, so I remember this as the summertime. I studied history and philosophy of science as an undergraduate there. And I read a lot about physics. And I think my next slide is a quotation from a physicist that has always meant a lot to me. James Clerk Maxwell is not as famous as he should be. Many people have heard of other physicists like Einstein, but Maxwell discovered electromagnetic radiation and explained essentially everything that you're now using with your cell phones. And he said this, which has meant a lot in the way I think about things. Thoroughly conscious ignorance is the prelude to every real advance. By ignorance, of course, he did not mean stupidity or ignoring facts. He meant a clear understanding of what you know and what you don't know. And so I thought I would take you to a short version of sort of my journey on trying to make beta cells, which involves a lot of mistakes and things where you didn't know, things that tried that didn't work. And then I'm going to move to try to convince you that we now think we have solved the problem and where we can go next. How to make beta cells. These are the sort of 4 ways my lab prosecuted this issue over a 25-year period. The first thing is when I learned about type 1 diabetes, the field believed that there were adult endogenous stem cells or progenitor cells. So there the idea is not unlike blood. Many of you will have been kind enough to donate blood, and when you give blood, you don't think you will be in permanent deficit for the blood because you have an adult stem cell in your bone marrow that makes more blood. People thought the same was true for the pancreatic beta cell. We spent a few years trying to find that cell and then determined that it doesn't exist, that there is no adult stem cell for a beta cell. In other words, if I were to use a laser and take the beta cells out of your pancreas, you have no capacity for making more beta cells. And that's important because if you're trying to cure the disease, you now know you can't depend on the patient to heal themselves. It just won't happen. That work was done with a wonderful postdoc fellow, Yuval Dor, who now leads a lot of diabetes work in Israel. We then looked at saying, well, if you can't make new beta cells, on number 2, can we replicate pre-existing cells? And there's still people working on that. My own opinion is that's unlikely to work. You might be able to get a round of division because there are some residual beta cells, but I don't think you'll be able to make enough to help a patient. We were able to show, and this third point is rather technical, by using a gene product called transcription factors to change one cell into the other, like say, make a doctor become a lawyer, something like that. That is possible, but it's not actually very useful. So we could turn the exocrine cells, the enzyme-producing cells of the pancreas, into beta cells. But to do that at a level that would be clinically relevant has 2 problems. It's very inefficient, and you're kind of robbing Peter to pay Paul. So it's not a good approach. Nevertheless, this is to show you that there are a lot of bad ideas that we pursue when you do discovery science that don't work out. The last thing worked, and that's what I'm going to tell you about. This is to take embryonic stem cells, cells that can make any part of the body, and turn them into functional human beta cells. So for those of you who don't do biology, I thought this is a very old slide. It must be 25 years old, just explaining to undergraduates what's a stem cell. So a stem cell has 2 interesting properties. One is it can continually self-renew. It can make an inexhaustible supply of itself, and that's quite unusual. That's not true for most cells. And secondly, if it's given the correct signals, as shown here on the bottom right, it can make all the specialized cells in the human body. And many people, many scientists, including me, thought this might be a good source if we could figure out how to tell the cells what to do. We call that control their fate, make them specialize or differentiate. And it seems so obvious to me that this is what you should do. And I'm going to take a little diversion here to talk about the JDRF, that I was naively shocked when everyone said, no, this is an immoral, unethical thing to be doing because you're using cells that are left over from IVF clinics. And in my opinion, were it not for the JDRF, I wouldn't have been able to pursue this. The JDRF's, Aaron will tell us what it's called. It's like its policy division or government affairs or something, arranged for me to speak with the president about enabling this kind of work to go forward. That was a long time ago. I don't remember when that was. But had that not happened, I think the US government at least would have said this is something that we shouldn't be doing. And I'm forever grateful to the JDRF for many things, but including that. In fact, Aaron, I think you still have that group. What are they called? Advocacy group, yes. So I thank the advocacy group. Thank you. So this was the idea. I like to show this slide because my lab always makes fun of it. They call this my skittled diagram. And it is a very simple-minded way of thinking about what the challenge is. On the left is an ES, or embryonic stem cell, and there's another version of this called IPS, called induced pluripotent stem cell, but these are these stem cells that I referred to. And the challenge was straightforward, is how to tell that cell to become, on the right, an insulin-producing beta cell. And the little purple dots coming out of the beta cell are supposed to represent insulin. And I remember when I started on this, I didn't know anything about diabetes, and my wife is really the one to make me work on this. I said, yes, I think I can solve that problem. And I want to emphasize the arrogance I had in saying that. She said, well, how long will it take? And I said, well, I don't know, maybe 3 or 4 years. And it took closer to 15 or 20 years. So remember that as I tell you about the future, is that my prediction won't be right, but it's a solvable problem. So we weren't the only ones to think this would be a good idea. Many other laboratories and several companies did. And I note here 2 companies, Novacell and Betalogics, worked on this, and they fused to become ViaCyte. And many of you will know that ViaCyte had, I think, a reasonable idea, which is they were unable to tell the cells to get to the end, so they got them to a pancreatic progenitor stage and showed that in rodents, some of those cells could continue their formation and become functional beta cells. And they were really the first to do clinical trials. And the readout of the clinical trials, unfortunately, is that that thing that worked well in rodents doesn't work well in humans. They've treated now more than 30 patients. And I think a fair conclusion is to say very few, about 3 or 4 of the patients, make a tiny amount of insulin, But unfortunately, most of those, let's call them green cells, ended up becoming alpha cells, not beta cells in humans. And there's no way they could have known that, and I admire and applaud them for having done all of that work. And as I say, it wasn't a bad idea, but like many things in science, it just didn't work. Our idea was to make functional cells that respond to glucose and secrete insulin, and use those for transplantation. How to make what we would then call a mature or a functional beta cell. This slide I have to take you through, even though it looks way too detailed, because it really represents the work of about 50 students over 15 years. And so in some ways, it's a sad thing to look at. Another thing, it's a happy thing to look at. It is fundamentally just like a recipe, a protocol for how to tell a cell what to do. And it turns out what we learned is that if you follow me left to right, an ES cell is an embryonic stem cell. We first figured out how to tell it to become definitive endoderm, DE. Hardly anyone knows what that means. But if you've ever caught a fish and you've gutted the fish, removing all of the guts is removing its endoderm. The endoderm is everything from your mouth to your anus, your lung, your liver, your pancreas, your stomach. So the first thing we tell it is what part of the body to make. Then pre-gut, then PP for pancreatic progenitor, then EN for endocrine, and finally beta cell. And the recipe, if you like, is to add these different factors. They're not named here. They're 2 factors and a different 2, et cetera, and how long it takes. And I just say that it's a complicated thing to do. It's probably like way more complicated than when you make your favorite chocolate cake or anything. There are many more factors, it takes longer, and it involved a lot of trial and error. And Aaron was talking about the importance of teamwork, and I want to just pause on that to say that in no way should you think this was like an idea I had and I knew how to do it. This was the result of the benefit of working at a great university with spectacular students who dedicated themselves at small pay and long hours, joining me for a long time. And I was trying to think about how to describe one special person, and I realized a good way to put it is that when you think about your children or your workers, in a sense, everyone is equal. But at some level, some people are more equal than others. And one of them is here in the room today, is Dr. Felicia Pagliuca, who was instrumental in leading our group. And she, I'm delighted to tell you, now runs the Type 1 program at Vertex. Felicia, would you just stand up so people could see you? Yes. And I'll say a bit more about Felicia as I go forward, but she was really one of the special members of my lab over this period of years. What I show you at the bottom is just a picture, an image for you to think, to demonstrate that the cells we make are really islets, not just beta cells, but they are indistinguishable from the islets in a person without diabetes. If you look on the bottom left, those are organ donor or cadaveric islets, and on the bottom right are the stem cell-derived islets, and they're stained for gene products that are essential for the production of insulin. The red is a factor that binds to the DNA and tells the DNA to make insulin, and the green is the insulin in the cytoplasm. So I'm not going to show you a lot of data. I'm just going to show you the kind of evidence that we've made cells that are what we wanted. In order to turn an embryonic stem cell into a pancreatic beta cell, one has to turn off about 2,000 genes and turn on about 3,000 other ones. So if you do the math on that, it's actually a very complicated process. And I'm not going to show you all that evidence for gene expression. I'm just going to show you what it looks like if you look at the cells in a microscope. So this is an electron microscopic picture. And you may not know that the insulin in your beta cells is complexed with zinc to make a crystal. That crystal is black, and it's shown here, crystallized insulin on the right. Each crystallized insulin is inside a little bubble, a vacuole, and there are about 100,000 to 500,000 bubbles in each cell. So you have to think about very small things. But the key thing here is on the bottom right is that the stem cell-derived beta cells make that vacuole with crystallized insulin, which is indistinguishable from the normal one. And cells could not do that unless they had turned on and off all the right genes. Here's one of my favorite pictures, which Felicia was involved with, and that is to say, can we make cells that properly respond to glucose and secrete insulin? Others have shown they could make cells that would make insulin, but they didn't respond to glucose, so they're not very helpful if they just have the insulin in them. In the middle, in panel C, are the cells that ViaCyte had used, which were unable to respond to glucose and make insulin, but could mature in rodents. On the left are the cells we make, and so I like to kind of joke about this, that there are 3 challenges with glucose, from 2 to 20 millimolar, that is the amount of glucose. And you can think of those 3 challenges like breakfast, lunch, and dinner. Each time you give more glucose, the cells secreted insulin. If you look on the far right, these are adult cadaveric or primary islets, and similarly you see when you add glucose at low and high, each time it makes more insulin. This was what really convinced us that we had succeeded, that we had made in vitro cells that could respond to sugar. I'm not going to show you the evidence then that we tested these in all kinds of animal models, all kinds of different diabetic mice. It's just not interesting. I'll just tell you it worked. Now we're faced with the problem that we have what the JDRF and others support, which is basic research. And Grant and Aaron talked about how, well, that's all great, but like, you don't cure anybody by publishing a paper or by going to a meeting. You have to make it and commercialize it. And so now I'll move to that next step, which is really out of the university. This is what the cells looked like, and now we wanted to make them at scale and think about how to treat people. And it's probably obvious to you that I don't know anything about that commercialization, and it's not like the thing I was trained in or think about. But I knew enough to know that, well, I was lucky to live in what I consider to be the world's best city for biotechnology. I mean, Boston is like, it is the center of biotech. In fact, my wife used to complain before COVID that when we go out to a restaurant, she said, everyone at the table next to us is talking about CRISPR or stem cells or something. She says, like, you can't get away from it in Boston. Boston is really a kind of biomedical mecca at the moment. So these are the cells, and I show you this slide because we make these stem cell islets which contain the alpha cell that makes glucagon and the beta cell that makes insulin, and they are the right size in the sense that they're the same size as human islets. And this is a funny slide to show you, and I'm showing it really to thank my wife. That's my wife's fingers there holding this. This is a little vial which contains stem cell-derived islets that were stained for insulin to make the insulin-producing cells purple. And the point of showing you this is how few cells, like the total amount there is about the size of a pea. So just think about how much trouble those cells cause. It's just the absence of those cells which are causing the disease. So these are human islets made, they're stained for insulin. And each dot, you can probably see some of the dots there, has about 5,000 to 10,000 cells because they're just like a human islet. So once that was possible, the question then is, how do you turn this into something that could help people? How do you make a product? And I don't need to remind us that universities don't do that. In fact, they almost think it's you shouldn't be doing that. And so I started a company called Sema Therapeutics, interestingly to me anyway, named after my 2 children, Sam and Emma. And I'm delighted that Felicia, who could have done anything in my view, she'd started, I think, at Harvard Business School, but decided instead this would be more fun. And so she joined me in starting Sema, and that company did a really good job. So Sema Therapeutics took this protocol and did at least 2 things. They improved it, like improving on a recipe, and they industrialized it and scaled it up and showed that they could treat larger animals, including pigs, and really get it to the point where you could now think seriously about making that product. Then there was another transition, and I'm not good at dates. I would guess it was 4 years later, where I met a former student and colleague, David Altshuler, who's the chief scientific officer of Vertex, and he just asked me how things were going. And I said, it's going great. And he said, well, maybe Vertex would be interested in that. And here I feel very lucky. This is by living in Boston. And Vertex decided that they would really like to join, as Aaron had said, in trying to cure this disease. It's a big company with considerable resources, And lickety-split, Vertex acquired or bought Sema Therapeutics. And Felicia then moved from Sema to where she is now at Vertex, along with many of the other people, many from my former lab and other people that had worked at Sema. And so now I'm going to switch really to talk not about my work, but let's say the downstream consequence of the work that I had done in Harvard, that what Vertex has done. So there's been a clinical trial with functional human islets by Vertex, and some of you will have read about this in the newspaper. I'm going to tell you public information and point to some things that aren't so public yet. But this was announced in October of 2021, success in the first patient. So what was actually done? The production of these stem cell-derived islets was scaled up to the point where you could make enough to treat a patient. And on the basis of calculations, it was concluded that, let's say, X number of cells would be required to cure a patient. The FDA, maybe unreasonably or reasonably, decided you shouldn't put in X, you should put in half of an X and go slowly. So the first patient was treated with what we calculated to be half of the required dose. This is from a press release by Vertex, and I'm going to show you a slide after this, but the main point I want to emphasize, so I'll say it several times, is that the cells work and they have made this patient insulin independent. That this person now makes his own insulin, does not require any insulin injections. You can look here on the left part, I don't have a pointer, but maybe I'll just walk over to it. There were undetectable amounts of insulin in this patient, that no measurable C-peptide. And 90 days after infusion of what's called VX for Vertex 880, is just these cells, he's making clinically therapeutic levels of insulin on his own. His blood sugars in the first 90 days were not only extremely good, but his daily insulin dose went from 34 units to less than 10% of that, 2.9. Now he's more than a year out. I think it's fair to say this was June 2021, so we're like 18 months out. And he does not inject insulin. I'll show you that in the next slide. And again, if you allow a kind of personal story, Felicia and David and others invited me to Vertex to show me these results. And Felicia might remember that I didn't really understand it because I was hoping, well, I'll actually tell you the whole story. Because it's patient information, I was not an employee of Vertex, so I was not allowed to know it. So David said, could you come on Friday and we're going to tell you some results? And I said to my wife, this has got to be bad. If it was good news, he would have told me on the phone. And so Gail said, when you get there, give them a talk, tell them we can't give up. It has, this is only the first. So Felicia shows me the results, and his blood sugars are nearly normal. And I thought, well, that must be the control. Like, where is the experiment? And she'll remember, it took a while for it to hit me that it actually had not, I was hoping for evidence for insulin production. I didn't dream that at half of the dose, he would be functionally cured. So I'm going to show you one piece of data from this patient. Yes, here. So this is kind of a complicated slide, so bear with me. On the left, you can see it says study day, and those numbers are negative, meaning that was before he got the transplant. On the y-axis, going top to bottom, you're looking at his glucose levels, and for those of you who read a glucometer, you see the enormous variation in his glucose in this patient. And I want to draw your attention to two things. One is the gray region, which is green, is what's called time in range, and that's what endocrinologists say we should aim for to make a person non-diabetic. I want to point out two things. Look at all the dark blue, which is very high, above 250 to 400, and probably some over 400. And then look under 70 at, is it kind of orange, or I can't quite see the colors here, but those are the hypoglycemic events, the dangerous low events that Aaron referred to, referring to his brother. Look on the table at the bottom, his daily insulin requirements of 34 units. The time he spent, let's say, in the required range was 40%, and his hemoglobin A1C was 8.5. He's even better now. This is at 270 days, and you see his glucose readings, taking no insulin, 99.9% in range, with a hemoglobin A1C of 5.2%. So those applause are really for Vertex, for Felicia and her team. I want to be clear. I'm not a real doctor. I didn't have anything to do with this. I'm just proud that the cells we figured out how to make work. Now you could say, whoa, hold on, that's one patient. I get that. It is just one patient. All patients will be the same. But in my view, this proves the point that it can work. There will be improvements required, but this will work. So I'm going to now summarize what public information from Vertex and then move to where I see the future going with the disclaimer that I've never been very good at predicting the future. I'm just going to tell you where I think we're headed. So at day 270, and he's now must be at day like 400 or something, receiving just half the target dose, this patient was insulin independent, and those numbers I've already told you. A second patient has received half the dose and has improved, but not as much. And there's new information which is not public on that patient. I think it's fair to just say there have been 2 more patients, and all I really should say is that they're doing well. Neither patient had any severe hypoglycemic events after 35 days. And as they often say in these sort of more formal technical things, the safety profile was consistent with immunosuppression. And I should remind you, and I'm not even sure I said it, the cells were delivered to the liver through the portal vein with immunosuppressants. I think, and I put this in blue, this is the first demonstration of insulin independence following treatment with a stem cell-derived islet therapy. And if I had any one take-home for today, it would be that I believe these cells can treat the disease. Okay, now we're going to move to the future. And again, on the personal side, I want to say why I moved to Vertex. I moved to Vertex because they are all in on trying to cure this disease. And I should have realized earlier that I spend too much of my time, like 40% of my time, writing grants and writing reports. And Vertex said to me, well, you can come, and I'm not supposed to repeat this publicly, I guess, but essentially I have an unlimited budget, and I can do what I want. And that, for like a scientific nerd, that's like that's like being a child in a sandbox, or like, it's just like the perfect thing, is I don't think about writing grants. Imagine that. Like, I don't have to explain to anybody what I want to do. I don't know how long that'll last. It may, but right now, I've been there about 3 months, and it's like I feel like a newborn. It's just wonderful. So there are 2 things I want to work on, and these are consistent with Vertex's plan. One is to gain complete mastery over the cell composition. So we now make islets that are very much like human islets, but we want to improve on them. We want to make more beta cells, fewer unwanted cells, and really end up with an islet that's alpha cells, beta cells, and another cell you probably don't think much about called the delta cell that makes somatostatin. And my opinion is that based on mouse experiments, those are the 3 cells that you need for perfect control. And we make all of those cells. We just want to be able to say, like, what would you call it, a designer islet that has the absolute right amount. And if we can improve that composition, we can reduce the number of cells to be transplanted. Then, as Aaron rightly pointed out, because he's always thinking ahead, is like, this is great, Doug, but we still have the immune problem. So, like, what are we going to do about that? And I'm going to talk to you now about eliminating the need for systemic immune suppression. The next slide I'll show you came from Sema Therapeutics, now part of Vertex, and it's one of the 2 ways to think about blocking the immune system. So let's just pause here and say, let's posit that the problem of making functional cells has been solved. Technically, that's not true. Scaling the manufacturing is not trivial. As I said, we want to control the composition. But let's just say that we now have an inexhaustible supply of cells. How will you provide those to patients when we know the patient has an immune system that wants to reject those cells? Let's also remember that that would be true for any cell. If you take these cells and put them into anyone, it's not just type 1 diabetics, because they're not self, they would be rejected. That's called allo, A-L-L-O, or other rejection, allo rejection. There are 2 ways the community thinks about preventing allo rejection. One is physical protection, encapsulation, which JDRF has funded for some years. Semma developed a very impressive device, an encapsulation device, which will go into clinical trials, and you can ask Felicia more of the details, but let's just say it'll be within the next year. They'll file an IND to do a clinical trial with this device developed by one of the best bioengineers I've ever met, Chris Thanos, whose picture is up here. And I'm just going to describe a little bit of the device. The device is made with a proprietary version of Teflon, and it has little chambers within it, and then a very clever design where holes are punched in the device that don't allow cells to go in and out, but let blood vessels go through. And so not all of the cells are within 150 microns of a blood vessel, which is the diffusion distance for oxygen. And in animal models, these have worked so well that it's now time to test it in humans. So I'm very excited about the next year's clinical trial with the encapsulation device. But again, as Aaron pointed to, first of all, I'm not a bioengineer. I barely understand these material sciences. I'm thinking about a biological solution to it. And Aaron pointed to this when he talked about CRISPR technologies and others, which the JDRF is supporting. And I'm not going to tell you details of the experiments. I'm going to tell you the challenge here to kind of emphasize that we don't know how to solve it. And that's what discovery science is about. But I think we have a pretty good idea of what we're dealing with. And that's here. So let's imagine that this purple cell here is an allo or an other beta cell. These are the cells we now make in unlimited amounts. What's going to happen when you put them into a patient that has type 1 diabetes? Well, first, the real answer is we don't know. We can make guesses about how diabetes develops in a mouse. No one has ever put these cells in a patient without immunosuppression. We don't know exactly where's the best place to put them. We don't know if they'll suffer from lack of oxygen initially, from hypoxia, or if the antibodies will bind to them and glom them up, or if T cells will, we just don't know. But I'm going to show you some of the things we think are likely to happen. Well, the first thing is we know patients have what's called a T cell, an immune cell, which will recognize beta cells and kill it. So we study how to stop that T cell interaction by deleting, using CRISPR technologies, some of the molecules that are on the surface of the cell that the T cell is recognizing. Second thing we know is even if we eliminate destruction by T cells, there's another immune cell in the body called a killer cell, a natural killer cell, and it will want to kill it. So we think we have another way of blocking that. We also know that there are these complement fixation molecules which will bind to the cells. We've got to figure out how to block those. The macrophages, which are like the garbage eaters of cells, will also find cells that look a little damaged and try to eat those. And then there are other inflammatory signals. What I want you to take from this is it's not a trivial problem. It's a complicated problem. And I was smart enough not to tell my wife how long this will take to solve. It will take a long time, and as Aaron said, a community of people. I'm betting on Vertex and our work there, but the JDRF is wise to say, well, good luck to you, but we need centers of excellence elsewhere, which will have other ideas to work on this. So I think that is the right approach. So I'm going to finish by saying what I think the way forward is. It's transplantation into patients with immunosuppression. That's been done. As I said, in the next year, I hope there'll be an encapsulation trial. But my dream is to use genetically modified cells that you can inject to a patient. So if I would sort of share that dream, imagine in another country, as Aaron said, a young girl is diagnosed with diabetes and comes in and her doctor tells her, well, I'm really sorry to tell you you have type 1 diabetes. It's no fun. It's not a good disease. But I have good news for you. I have in my freezer some cells which I'm going to inject into you. And maybe by then those cells will last for more than a year. So I'll inject these cells. They'll control your blood sugars and you're good to go. So I'll see you in a year. I don't know if that'll happen in my lifetime, but that's my vision of what we should do. Is find a way to restore the body's natural ability by making nature's solution and figure out how to inject them into patients. I appreciate that it's equally or more important to work on closed-loop systems and algorithms. I just don't think about that. This is the thing that I want to do. So number one, in my view, has been done. Number two will start. There are too many people for me to really thank. In black are the people who are in my lab still at Harvard. In blue are my colleagues at the JDRF Center of Excellence in New England. And I'm now delighted to say I work at Sema Vertex on this problem. And the bottom are the grants, which I'll never have to write a grant. The Howard Hughes Medical Institute, the Harvard Stem Cell Institute, the Pick Our Foundation, the NIH, the JDRF. I'm very grateful to them, but they'll never see another grant from me, so that's good. So thank you for being so patient, and with Aaron's help, now I'm glad to answer any questions. Thank you.
Pass around mics to folks who want to ask a question. Doug, we'll go right to Emily.
And I can project.
Well, we want to be able to, they told me to make sure folks have mics.
Okay. Oh, my goodness. Yeah, that is loud. It was fabulous. Thank you. I have really a number of questions, but I'll start with just one. How long, you mentioned that you would think that the cell would be lasting a year in your hypothetical situation. So how long do you expect the cells now to be lasting? That one patient is at 270 days. Would you expect to re-inject with another batch, or how do you mark it?
When I say expect, I'm going to just give you my intuition. These are like newborn baby cells. They are much better than cadaveric islets. Cadaveric islets are isolated from a dead person with enzymatic digestion. They're not the best prime material. I think these are like newborn cells. So if you ask me if the immune system can be blocked, they will last for the lifetime of the patient. Now, that's probably unrealistic. We already know they last a year at half the dose. So they're going to last more than a year in the presence of immunosuppression. Consistent with your question would be then, well, Doug, how long will they last in the device? We don't really know. We know how long they last in pigs and other animals, but that'll be really important. If you allow, I might give you a personal answer to that. I told my children about this encapsulation thing, and my son Sam, who's only known diabetes at 6 months, he said, well, yeah, let's wait a couple of years and see if it works. And my daughter, who had been pregnant dealing with diabetes, said, I am so sick of this disease. I want to be in that trial. I don't care if it only lasts a month. If I could have a month of it, I would do it. So it's a long answer to your question, really saying I don't know, but I think they will definitely last more than a year. Is that fair?
Yeah, no, that sounds good. And back on a more basic science question, because you mentioned that we now know that adult stem cells won't actually do any work of making new beta cells. Did I understand you say that right?
What I said, which is correct, is that there's no evidence that an adult stem cell can replenish beta cells in people. What you might be, technically your question would be, could you get a cell from an adult, turn it into a stem cell, and then turn that stem cell into a beta cell? The answer to that is yes. That's called an iPS cell. But that's not the patient doing it on their own. You have to get the cells out and then go through the whole procedure I described.
Okay. So when, at one point some years ago, we saw a study that pregnant women end up with more beta cells, more greater beta cell mass.
That's true. And those beta cells have divided, and we and others have tried to figure out what that signal is, but it hasn't been determined.
Okay, because I thought that was the basis for the hope that a person could sort of cure themselves if you stopped the attack. But
I would say hope, yes, but evidence, no.
No. Okay.
Yes, right.
I don't mean to hog the mic, so. Okay, Jordan.
What is your theory as to why the first patient responded more fully than the second patient?
I'm glad you said theory. We don't really know, but I would say that it probably has to do with the person's insulin requirements. And there's some evidence, which I don't think is public, that the immunosuppressants given to the second patient were delivered in a different way. And so then, but I know enough about clinical trials to say that, first of all, humans aren't mice. And secondly, there's huge variation, as you may know, several diabetics. Some diabetics require more insulin, others not. Every patient is going to be different. And really the challenge for Vertex now is to figure out, can we find a dose that will treat 90% of all patients? So we don't really know yet. In fact, one might say it's very lucky the first patient did so well.
Well, and maybe Felicia wants to comment on this. I asked that same question when I was at Vertex, and the other question, the corollary to it to me is, well, the dose, are there risks for increasing dose? Like I was trying to get my arms around what FDA was thinking. Like, okay, you put those many, we're talking about a tiny amount of volumetric cells. And I don't know, Felicia, if you want to comment on that, but ramping up the dose seems to me to come with not a big risk profile.
I'd be interested to hear what Felicia says, but my view is you should give more than the required dose. Because the cells auto-regulate, and it's a tiny volume. The FDA doesn't seem to understand that yet. This isn't like injecting insulin. It's an auto-regulatory cell. Felicia.
Yeah, I think that point is, ooh, that's loud, exactly right. I think the treatment paradigm is so different than what you think about with exogenous insulin injection, where there's no ability to titrate exactly the right amount of insulin to the need in terms of glucose levels. And so we really think that you have the ability to increase the dose of beta cells you deliver without significantly changing the safety profile of the cells, because one of the other critically important outcomes of these early days of the clinical trial is the data that shows that these cells are truly self-regulated in the way we hypothesized, in the way we saw in a petri dish and in an animal model. So we see that those insulin levels go up and down in response to changing glucose. So demonstrating that self-regulation capability in these early patients in the trial was critically important, and I think sets us up to be able to escalate to the kind of doses that will get everybody where we'd like them to be clinically.
Yeah, and to follow on really the first question about longevity, my view is you should put 10 times as much as you need. Because in an animal, we put 10 times as much, and the animal is fine. And that means if the animal is slowly killing them, they'll just last for 10 times longer. The FDA doesn't accept that view. Maybe the equivalent here, and there will be a clinical trial here in England, I think, at some point, and maybe that the FDA equivalent will be more open-minded.
And that, just to be clear, we talk about our advocacy group, and we've really built out our health policy team, working with Vertex and other companies in the space to get to better clinical trial design and criteria. It's been a challenge, and Felicia and the team and I talked about this. To recruit for this trial, the bands of recruitment are very narrow, the dosing, and our team is in full tilt trying to improve that process. We've got a bunch of folks up here.
Thank you. Thanks, Doug. I'm intrigued by your early comment about taking 15 years to achieve a 4-year goal, and now your later comment about what you can achieve with an unlimited budget. If we'd found you 15 years earlier and given you that unlimited budget then, how much more quickly could you have achieved what you then took 15 years to achieve? Because what we do to scientists like you is ask you to reapply for your pathway to success repeatedly through grants. It's a systemic problem around the world. What would have been different if you'd had that unlimited opportunity earlier?
Well, I would say the biggest impediment to our progress was the controversy over using embryonic stem cells. So I spent more than a year developing the cells in the basement at Harvard because it could involve no federal funding. And then we sent the cells out to everyone in the world who wanted them for free. That slowed us down. The other thing that slowed us down is bad ideas. I might even say stupid ideas, things I wanted to try that didn't work. So that makes it harder to predict. But you're asking a very direct question. If I had unlimited funding, I would say at most it would have reduced the time by half, but I never would have got to 4 years. That was just my own ignorance of how hard the problem was. Yeah.
We would take half. Alana. Who we have in the front?
Couple of questions. These initial 2 patients, you injected the beta cells into the liver, is that what you said? And do they then settle in somewhere in the liver? Is that, okay. Okay, so understand that model. With this matrix that you're creating, where will that go and where will those sit in the body?
The present plan is to put them in the abdomen, under the skin and first layer of muscle. The one advantage of the liver is, maybe this is too detailed, but when the pancreas secretes insulin, 50 to 80% of it is taken up by the liver. The liver is the main place it has to go. So putting the cells in the liver is a good idea. We don't know how well it'll work with a device that's not at the liver, but in no instance does anyone think about putting the cells back in the pancreas. So that they will not be, and that's, you could say, one of the good things about diabetes is you can inject insulin pretty much anywhere. And if you're thinking of heart disease or brain disease with stem cells, it's not going to do any good to put your heart cells or your brain cells in your abdomen, right? So there we're kind of lucky that we know we can provide the insulin elsewhere in the body.
Okay, so basically somewhere under the skin in the abdomen is what you're thinking? Or,
Right.
Yeah.
And that's something, Alana, that we, the team and the research that we're doing are looking at encapsulation in different, like one is in the omentum. There are different groups looking at different options. Still an open question.
And one last question. In terms of the stem cells, are those being just grown in the lab from a basic starter stem cell? Is that what we're
That's the
looking
amazing thing about stem cells is you could, in theory, begin with one cell, and that can be enough for centuries for the whole world.
Wow.
I mean, that's why I can't understand why every undergraduate doesn't want to work on this. It's so interesting.
Yeah.
Like, there's so many
Thank
things
you.
you could do.
I
Right.
say Claudia and then Elizabeth.
Thank you. Maybe could you comment on the progress that the scientific community has made on immunosuppression compared to where it was 20 years ago? Because, you know, we are seeing more cell generation. How far have we come to immunosuppression? And is that a possibility to continue at the existing immunosuppression, you know, drugs
Yes,
now?
it's a great question. An experiment that would come to mind is we give immunosuppressants that are used for organ transplant. And the question is that right for islet transplants? The fact is they work, but maybe they're more than are needed. But I think your question more broadly, what progress has been made? I would say one of the biggest advances has been in cancer immunotherapy with so-called CAR T cells, which show that you can actually manipulate the immune system with cells. And there are people here from Provention who work on, I can't pronounce the name of teplizumab or whatever. This is an anti-CD3 antibody. That's also a real advance to show that you can dampen the immune system in a very productive way. What would be really interesting to know is if you could identify all of the things that T cells and antibodies recognize on a beta cell. First of all, are all patients the same? This would be lucky. If all patients were the same and there are only 6 important things, we could have 6 versions of teplizumab and then say, that's it, we're done. On the other hand, if every patient is different, systemic immunosuppression won't work commercially because you can't do it patient-specific. But that's like an exciting puzzle. How many different ways are there to get autoimmunity?
Some of you have heard me tell this story, but I had this discussion when I visited Felicia and the team up at Vertex, and David, who Doug referenced, who heads up science at Vertex, he said to me flat out, would you do this? And of course, this requires immunosuppression. And I thought, huh. And I remember coming back from that meeting, I flew back to New Jersey, and I was telling my wife how excited I was about what they were doing and how they asked me that question. And she said, well, what'd you say? And I was like, I think I would do it. A lot of people are surprised, and she goes, you don't want immunosuppressants. And I said, kind of don't want diabetes. And, you know, it sounds a little glib, but I think there's a lot in that. One is I'm a big believer, when this comes back to the policy, that we need to work on FDA, and we've done this for years, Cynthia Rice and the team and I, on the living with diabetes today still is very difficult and risky. And providing that patient voice for, like having the FDA tell me that I shouldn't be able to do this, to me is not the right answer. And allowing for more flexibility for if there's a safety profile that we understand, that we should allow people to make that decision. Would I do it if my child, God forbid, got type 1? Probably not, but I'm older now. And I think there will be a number of people who will benefit from a first product that does require immunosuppression. That's not our goal. As Doug said, we are working full tilt to remove the need for immunosuppression. But there are a lot of people who could benefit from a therapy like this right now. And I think a very important role that we'll play, we, JDRF, in the community, is educating clinicians and regulators that there's still a huge unmet need for people with type 1 diabetes, and that people with type 1 diabetes need to have a voice at that table. Oh, Elizabeth, sorry.
So I have 2 questions. We're so grateful that Vertex and many other industry partners are investing in cell replacement therapies. But you alluded to questions unanswered, and that's a lot of what the foundation's research is focusing on and the fund's research is focusing on. Can you comment a little bit on some of those questions that still need to be resolved so that we can get through a first, second, and third generation product?
Yes, that's a very thoughtful question. So I would say, first thing to think about is, should one try to dampen the immune system in the patient so that it wouldn't recognize any foreign cell? That's called systemic immune suppression. And many people like that idea. They could be right. They like the idea of using T regulatory cells for that. I don't think it's a good idea, so I wouldn't do that. I think we should take advantage that we have a defined drug product, something we can make, and modify that so that it is opaque to the immune system. And so if you ask me, like, what areas, which I think you're asking me, do I think are ripe for giving informative results for this, I'll remind you that cancer cells have figured out how to block the immune system, and that one of the most basic things in life that we never think about is when a woman is pregnant, she should be rejecting the baby inside of her because it's not self. There's an individual called the sperm donor, or otherwise the father, whose antigens are inside of her, and if you took skin from the father and transplanted it to the mother, she would reject it. What's that all about? So I'm really interested in the maternal-fetal interface as a clue to how we might make beta cells that, if you like, look like the fetus or look like a cancer cell, even though they're not either one of those, using CRISPR technology. So you're kind of asking me, what would I bet on? That's what I would bet on. But I hope other people have other ideas. You have to, discovery science means you have to pick some idea and then really squeeze it.
And that leads really well to my second question. Can you comment on the acquisition by Vertex of ViaCyte and what you think that might lead to in terms of CRISPR and advances in that way?
Well, it doesn't have anything to do with CRISPR, in my view. I think it's kind of doubling, my view is that's Vertex doubling down the same. They want to get all the resources they can to solve this problem. They're manufacturing facilities, and as I said, ViaCyte and its previous companies must be 10 years of experience with clinical trials. So there'll be valuable information about trial design. They have collaborators in Canada and elsewhere. I hope you're not asking me to comment on the business part, which I don't understand. So just, I would say it's a good sign that Vertex did not do this as one try and say, if it works, well, we'll decide. Vertex is completely committed to this problem.
The other thing that I'll just comment on, Elizabeth, that we see at JDRF is, as you know, we believe that we want to push on fields. And I think the acquisition of ViaCyte by Vertex puts immense pressure on the other companies to ramp up their games and get moving even faster.
Yeah, a mean way to say what Aaron is saying is if you're making money selling insulin, you better pay attention to this.
So we have Katie and then Mike.
And maybe David Panzier. But just a quick question, maybe you can clarify for this audience. When you talk about immunosuppression in the context of beta cell replacement, there's both the rejecting it's not your cells, and then there's the fact that people with type 1 are destroying beta cells kind of naturally. And so maybe you can comment on those two aspects of the immune system and how you see that evolving, or how does that play out with this type of therapy over time?
Right, that's a good question. So let me remind us all that we're transplanting not just beta cells, but alpha cells and other islet cells. And so the autoimmunity isn't really the main problem. It's the alloimmunity. It's that these cells are foreign. But what one does not know is, imagine you solve the alloimmunity problem, that being a non-self was the issue. Would the autoimmunity that's there be a problem? Nobody knows the answer to that. And in fact, we don't even have really good models, animal models for that. We're only going to learn that in the clinic. And maybe the device will work. I skipped over the device. I personally think this device is extremely clever and works very well in pigs. So I'm excited to see the results of the device in people.
Mike?
Over here, Dr. Melton. So when I was in management, stating the obvious was very important. So I'm going to state the obvious here, Dr. Melton. As a fellow father of a son with type 1 diabetes, I just want to thank you for your brilliance, your dedication, your hard work over so many years, and for giving us all so much hope. So that's really what I want to say to you. Thank you.
Yeah, thank you for that, but I feel that you all here are doing the same thing. Any parent who has a child with this says, what am I going to do about it? And so I appreciate what all of you are doing to try to solve this problem. I have just a kind of very narrow area of expertise. I couldn't do fundraising, I couldn't do any of the things Aaron does, so, but thank you for your comment.
Matthew?
A two-part question. Well, of course, I echo the earlier comment, but a two-part question. You showed that it takes 180 days or whatever for the cells to get fully insulin, fully glucose reactive. Why is that? What's the theory about that? Why don't they work day one 100%? And then second question is there any possibility that this becomes more like a smart insulin concept, meaning you don't solve the immune problem, but you just inject new cells once a week and you keep fighting the immune system by adding more cells? And I don't know how long it takes the cells to ramp up and how long it takes the cells to die.
Yeah, great question. So we don't know why it takes them time to ramp up. We do know the cells work the day they were put in, before they went in. The present idea is that you have to wait until the blood vessels around the liver come in. They infiltrate the islet like a spider web. Like all of those cells I showed in that picture, each cell gets contacted by a blood vessel. And so they're right next to oxygen in that. And we guess that it just takes the body that much time. It can also be that the lack of oxygen means the cells say, look, I can't breathe, so I'm shutting down, right? Like they just are dormant until they get oxygen. So I would say oxygen, or what's called vascularization, is the #1 idea. Second point, you're right, is, well, what if you can't deal with the immune system, but you would just keep injecting cells? Well, in the absence of immunosuppression, there's good evidence to believe the cells would only live for a couple of weeks. And so that's really not practical. But in the presence of immune suppression, this is what I think Aaron was referring to. What if you had to go on immunosuppressants and you had to get cells injected every few years? I'm not so sure that's a bad idea if you are, as my children would say, relieved from the daily craziness of worrying about my monitor, my catheter, my pump, what am I eating, what's my blood sugar. The first patient says he doesn't think about that anymore. I think that the value of that is underestimated. In other words, how about this? Even if it does not improve hemoglobin A1C, what if it just relieved you of the daily management? I think that's an important product. Did that answer your question? Yeah.
John?
Again, I echo the comments about your journey here and you staying hyper-focused, and I really want to thank you for that. and I think that will pay off in spades in the future. But as this journey continues, and we'll continue to work on the immune system and the response and trying to deter that, this second part that you talked about, using encapsulation and maybe the lessons from ViaCyte that worked well in the animal models, and now you're seeing the device in Vertex working well in pig models. What are the lessons that have been learned maybe from the ViaCyte experiments, and what are you doing differently in your macro encapsulation? And then just as a quick follow-up to that, has there been any work done in the micro encapsulation?
Yes, to the second, but I'll start with the first. ViaCyte's idea about, let's call it macro encapsulation, was not a bad idea, but it didn't work. They thought you would want a device that induced fibrosis to bring fibroblasts and blood vessels there. It turns out that kind of gloms up the whole thing and prevents oxygenation. So their device did not work. Didn't work well in animals. In rats it didn't work well, and it hasn't worked well in people. So you might know that while they use a device, they drill holes into it, not to let blood vessels through, but just any cell can get inside that and kill the cell. So the device with holes requires immunosuppression. Vertex's SEMA's device is a completely different material and different design. That's why I think it will work. Then microencapsulation, many people will know that JDRF has funded this for years. In general, more than half of that involves taking seaweed extracts, alginate, and putting them around cells and thinking you can protect them. That provides some short-term protection. In my opinion, the principal problem with that is it's not so micro. And so if you calculate the volume of cells you'd have to put in that are alginate beads, for example, it's somewhere between 500 mils and a liter. So it's a big, big volume. And then if you have to do that again, so you know how much a liter is. That's a huge volume. The thing that, remember that little vial I showed? It's the size of a pea. So I'm not a big fan of the microencapsulation, but I've been wrong about so many things. I don't want to stop people from doing that. I say, yeah, more power to you. Go, if you're starting a company to do that, good luck to you. Get going, yeah.
David, did you want to make a comment, question?
Sure. So, Doug, I know that you're obsessed with your little area, and I'm very, very grateful for that. I am obsessed with improving care because, as you said, you're not so sure you're even going to see this in your lifetime, and I'd like to think you're going to be around for a while. So, I remember back when Novacell was around, like, these cells are the best CGM you can have. There's no day one, they last for a long time. And I remember Novacell was looking at creating a CGM-like device using these cells. I don't know whether that's in the cards at all with Vertex or thought of. I haven't heard of anybody else working on it, but I was just curious I know it's not your thing, but
Well, thank you, David. I think if I understand your question is, could you use these cells, which read blood glucose every millisecond, in some kind of device instead of a continuous? I think you could, but I don't want to work on devices. But yeah, ask Felicia. Well, good. You've been a great class. Are we done, Aaron? Because I'm going to
we have Leah.
I have a quick, probably very basic question. You talked about turning your attention to mastering the composition of the cell. What does that really mean? And would there be a perfect composition? You talk about the fact that people are individuals. We have 2 kids with type 1. Their type 1 presents itself very differently. How they react to things is very different. Will there be a perfect cell that you'll create, or what does that mean, mastering the cell composition?
No. Thank you for the question. What did I mean by that? I would say think about a cell that doesn't exist in humans, a super islet. Think about, like, I forgot what the name of those Marvel movies are, where a person has a shield or some. We can make a cell which doesn't exist in humans, which will block the immune attack. We might even be able to make a cell that will live forever. So these are kind of crazy ideas, right? But I actually think it's good to go to the extreme with the idea and then whittle it down to what you can actually do. So when I say designer or mastering composition, it would be that at Vertex we would say we want to make islets that are 60% beta cell, 30% alpha cell and 10% delta cell. That islet does not exist in humans. Human islets have other cells in them. They have a cell, an epsilon cell, that secretes ghrelin, which controls appetite. Diabetics don't have trouble with appetite, so I'm not, you see what I mean? So we make something that doesn't exist. It's informed by natural biology, but it's a supercell. Yeah, so that, you've been such a good class, I want you to go home tonight and write an essay on what is a super islet going to be like, and then send me your idea. Yeah.
All right, well, I am getting the signal. Doug, I'll echo Mike's comments. You're an amazing champion, a great friend. You've been fighting the fight for a long time now. We all are tired, but I see light at the end of the tunnel. I believe it's going to happen. I believe this team and our community is going to help us get there faster, and I can't thank you enough for all your
thank
leadership.
you. And, I mean, I'm not tired. I'm excited. I think we're at a really great part. We're on the path to a cure. Yeah,
Awesome.
but thank you for all your
Thank you.
help. Yeah, thank you.